Today we will present you our project in Metabolic Engineering course. This is our team. I'm Lalo, Nati, Miguel, and Laura. And our project is about microbial overproduction of hyaluronic acid in Escherichia coli, straight K12. Well, what is our project about? Our project is about produce hyaluronic acid that is a linear polymer made of repeats of two monomers that are a disaccharide. The monomers are beta-1 4-glucuronic acid and beta-1 3-N-acetylglucosamine. Next one, please. Okay, as you can see, this is the principal reaction made by hyaluronic acid synthase. That the reaction is about binding the acetyl-D-glucosamine and the glucuronic acid to produce a chain that is the polymer.
Okay, we are going to explain you several strategies to overproduce this hyaluronic acid in Escherichia coli. Among the applications that the hyaluronic acid has is in cosmetical applications. It has the ability to retain water, and this is amazing because then we can use it to increment the collagen in the skin, and this way to regenerate it. Also, in the medical applications, it is used in degenerative joint diseases and also osteoarthritis. And among other applications is the pharmaceutical industry and the nutraceutical industry. The hyaluronic acid is usually obtained by two ways: using the natural resources. First, we have the rooster combs, and the second way is by the umbilic cord of the human babies. So the disadvantages of obtaining this hyaluronic acid from these two sources is that it is limited by the tissue they have. If there is no a lot of rooster combs, then we won't have a lot of hyaluronic acid. This also the medium is expensive, and it presents pathogenicity issues when transferring the hyaluronic acid from the rooster combs. Then, because of these problems, then the scientific thought of another way to obtain the hyaluronic acid, and this is by Streptococcus subdermicus. The disadvantages of this source is that it has a lot of viscosity produced by the poor mixing and low transfer of oxygen. The second disadvantage is that it is a competition between the production of the hyaluronic acid and the cellular growth, and also it can be formed the lactic acid in the cell, so this inhibits the growth of the microorganisms.
Also, as we know, Streptococcus epidemicus is a pathogen, so it's not a good idea to use it for industrial scale process. Well, what is the significance of this study? As we know, the worldwide market of hyaluronic acid is increasing every year. In 2012, the global worldwide of this market was $1 billion. And we project that this amount will be still increasing during the years. Nati told us about some application. The first market product that people designed was Pfizer in 1979 developed Hyaluron that was surgical aid for some illness in the eye, maybe for cataract extraction. Then they use it for a viscoplastic supplement for people that have osteoarthritis, and then is maybe the most popular use for hyaluronic acid nowadays is for dermal filler, because as we know, cosmetic products and cosmetic industry is gaining market, and a lot of people spend money in that aspect.
So, well, what is the proposal of this project? As my teammates also talk about the high hyaluronic acid, we want to overproduce this hyaluronic acid in Escherichia coli strain K12. So, what is this good idea? This slide, please. As Lalo and Natalia also said before, the hyaluronic acid could be extracted by umbilical cord and also could be produced in a Streptococcus strain. They also said that if you extract umbilical cord, you waste that, and you can use umbilical cord because it has stem cells. You can use it to treat several diseases. So also it was the concern about the pathogenicity in Streptococcus. So we don't want to overproduce that hyaluronic acid in that strain. So we propose to use Escherichia coli, and we need to make some improvements in that strain to make more desirable this project. So what we need to do is to lower the cost of the media. We also need to increase the production or produce hyaluronic acid. And as we are using Escherichia coli, we can forget about the pathogenicity thing.
Okay, so as the members of my team said, we have 5 strategies, and the first one is to explore the biosynthetic features of hyaluronic acid in a gram-negative strain to express essential enzymes in the pathway. This is by taking advantage of E. coli's fast growth and rate, and that's why E. coli is frequently used in genetic engineering. So what we want is to analyze the analogy in Escherichia coli and Streptococcus genes, and these genes are the ones that encode for these 3 enzymes that are the essential enzymes in the pathway. So by using BLAST, we did the alignments to see the percentage of match in those genes. So the first one is the UGD and HASB that encode for the first essential enzyme, and it has 83% of match. The next one is GAL-F and has C, that is for the second essential enzyme, and it has 72% of match. And finally, the third one, that is GL-U and has D, and that has 94% of match. So all 3 have more than 50% of match. One gene from the gene with the Streptococcus and Escherichia coli. Then the second strategy was to insert Has genes in E. coli. This to overexpress the overproduction of hyaluronic synthase to increase the hyaluronic acid. It has been reported in papers that the time that if you increase the time the hyaluronic synthase is with producing the hyaluronic strain, then you will have a higher molecular weight. And why does this matter? It matters because, for example, it was reported also that if you have a molecular weight between 0.5 and 1 megadaltons, you will have 1 doses of 5 weeks. That lasts 5 weeks. But if you have a molecular weight that is among the 3.5 and 6, then you will have by 3 weeks. So with this shows that if you, this is just one application, but it has others. So if you increase the molecular weight, then you will have a better among applications for this hyaluronic acid. The objective is to reach and overcome the molecular weight of hyaluronic acid, and it's also important because if you obtain the hyaluronic acid from the natural resources, as we said, the umbilical cords or the rooster combs, you have a high molecular weight. But if you obtain it by fermentation in the Streptococcus, you will have a lower molecular weight. So by this way, we can overproduce the hyaluronic acid in E. coli and also have the advantage of having a molecular weight. Well, to do this, we will use this vector that is PET28. And as you know, in genetic engineering, it has a T7 promoter. It's good because it produces a lot of what we want. That is the sequence. Of the enzyme, the hyaluronic synthase. Also, we found in this, this author said that if you overproduce E. coli with different conditions at different times, here you have the line of what is supposed to be the hyaluronic acid. and it has a bigger molecular weight than if you use it in conventional ways.
Well, another strategy is, as we know, for increased amount of a product, we have to increase the amount of carbon that goes to a certain pathway that is the interest pathway to produce that product. So we propose to improve the performance of phosphotransferase activities that we know that is the enzyme that will phosphorylate the carbon, well, the glucose that will go to inside of the cell. Well, as we know, and the teacher repeated us a lot of times, maybe this will be the first and the principal step that we will never forget about to start metabolic disturbance. Well, the second strategy is to find a gluten-sensitive enzyme to solve the problem that is the local negative feedback, because we check in Metabolic that this pathway is a negative feedback controlled by the presence of N-acetyl-glucosamine. So if we find a mutant enzyme that is insensible to the concentration of this substrate, we can keep increasing the amount of this product that is essential for the production of glucosamine. That is one of the principal substrates for the construction of our product.
So the third strategy, we talk about the cost of the media, that it was expensive. So we think about a component that we could use, and it was cheap. So we thought about glycerol. Glycerol is a second compound produced in the biofuel synthesis. So it's a waste, and the cost is very low. It was 1 to 8 cents per pound of glycerol. That depends on the quality of the product. So that's a good candidate of a alternative carbon source. So what we need to do is to use glycerol as an alternative carbon source. The strain already have the metabolic pathway. Well, I'm going to explain how this works. This is the normal pathway that the strain do consuming the media to reach or find a product. That is one of the two monomers that produce the polymer. It's right here. So what we want to do is to take glycerol from here. Yeah, from here, sorry. From here, take the glycerol and add it to the pathway here, right? In some intermediate, that is the glyceraldehyde. So we save all the carbon flux going through all that pathway. And we can do less reactions to reach one of the monomers of the production of hyaluronic acid.
As I said before, we use BLAST as a bioinformatic tool for our project, and we also use Metabolic. BLAST find regions of similarity between different sequences, and that's what we did with E. coli and Streptococcus sequences for the genes. And we can also compare protein and nucleotide sequences in other databases. And for the pathways, we use Metabolic, and we know that Metabolic has 2,453 pathways from 2,788 different organisms. So we can use this in order to achieve our goal in doing the disturbance of the pathway.
So well, we found in other papers that also do the same as we want to do in other strains. We found 3, Lactobacillus lacti, also Escherichia coli, but different strain, top 10, and K5. So in these works, for example, in Lactobacillus lacti, they introduced 5 genes. In the Escherichia coli top 10, they just introduced 4, and K5 just introduced 1. This is our interest gene, has A, that we also are going to introduce in our strain. So these are the results of that papers. They produce approximately 0.5 grams per liter. So that's what we are expecting to, or that's the minimum production that we are expecting to produce with all the metabolic engineering that we are going to do.
Well, the conclusions is that I will remind you the strategies we used. That first Laura said about the alignment between E. coli and Streptococcus of the genes of interest. Then the second one that I explained, it was to insert the hyaluronic synthase genes in E. coli these to overexpress the sequence encoding this enzyme. Also to overexpress the reactions of the proteins, of the upper reactions. Also to improve the performed phosphatase activity. And then find a gluten insensitive enzyme and change the carbon source to glycerol from E. coli culture media. All of this with the objective of overexpressing the hyaluronic synthase, improving the amount of substrate, decrease the cost of production, as Mike said about the carbon source, also to increase the molecular weight of hyaluronic acid change, as I said about the several applications that a huge molecular weight of hyaluronic acid has. Also improve the amount of carbon that goes